Okay, so our next speaker is Heinz Koeppl, who's going to tell us about statistical inference of single cell and single molecule dynamics. Assuming that
Yeah, thanks a lot. Thanks a lot for the invitation. So I'm going to tell more or less two stories. The first one being working on single cell data. So there will be a lot of data in this talk, which actually has no real relation to space. But in the second part, where I'm talking about our single molecule data, I think this very well relates to to space. And so I'm also co-affiliated with the Department of Biology, and so we can we're running a small wet lab there and trying to do some experiments. Okay, so this is so this died off now. Okay, so this is joint work with students of mine and also Christoph Zechner, who was a student of. He's now in Mustafa Kamash groups, and Dan Larson from NIH. So what we are interested in is, in general, getting somehow a more realistic model for a network of interest. And we are mostly talking about population type of models. So I'm not an MD person, so I'm more on the higher level, talking about chemical kinetics. And so most people are considering, so maybe I get this to work quickly, let me see. Yeah, perfect. Okay. So what we often do is cutting out our network of interest and studying this in isolation, but this is normally never the case that we have the clean data only of the network itself, so it's always embedded into some kind of random environment. It is the molecular context of the cell. And I believe that we somehow have to account for this if we build stochastic models or any kind of model of a cellular process that one is interested in. So we looked around and tried to come up with some inference algorithms that take account for this, what people call extrinsic noise sometimes. Okay, so any kind of influence of the molecular environment on the target network that you're actually looking on and also measuring. Okay. Yeah, and there is a lot of evidence that this is the case. So if you take single cell data, then there is large cell-to-cell variability that cannot be only explained by the intrinsic noise, by the stochastic dynamics of the system. There is also a lot of cell-to-cell, so cellular heterogeneity or influence of the extrinsic environment on the target network. Okay, so cellular heterogeneity and this random environment is more or less the same thing. If you think about it, you can explain both within. But I think a very easy example is that you have, for instance, your gene expression model, as we saw in the previous talk, and then you have, if you want to model the protein levels, then a player that is not explicitly modeled most of the time is the number of ribosomes that actually do the job. It's just encoded into a static translation rate, but the ribosomes are different from one cell to the other, and they are also fluctuating. So this would be a simple example of a random environment that is definitely influencing the dynamics of your network, right? So this has been also quantified in terms of these dual color experiments or dual reporter assays. Okay. Okay, so the key questions I want to address today is, can we build some type of equivalent model of the target network that we are interested in that behaves as if it would be still embedded into this random environment, right? We don't want to always keep on modeling the whole cell, these large thousands of equation type of models, whole cell models. We want to keep our process of interest, but we somehow want to account for a generic effect of the random environment. Okay, can we build such a model? And then clearly this also has an implication to simulation, because then you don't need to simulate the whole system anymore, but just the subnetwork, if you somehow know what effect the environment is doing on the thing. And then using this framework, can we then answer the question of can we decompose this variability that we see in the single cell data into these two components, one variability coming from the variability of the random environment, and one just really due to the stochastic kinetics. Okay? And then the last thing where it really goes to space, let's say, is can we use this framework to then do something very concrete in this case? Can we infer the RNA polymerase position from intensity traces of flavored nascent mRNA molecules? Okay, so there we have some single molecule data for this. Okay, so it's mostly all about inference. Okay, so the generic way of accounting, or a simple way of accounting for a random environment, is to build a joint model, as I already explained to you. So let's assume that we have a model of a random environment given by this red network. So we generate a realization of this process, and then we inject it into our target network, and then we somehow simulate the target network conditional on this particular realization of the random environment. And then we have our traces, our abundance traces, that then somehow resemble the whole heterogeneity that we see in the single cell data. Okay? So that's somehow so we want to build a model that, if I just keep on drawing from this, I generate such traces, okay, without building the joint model. So here you see, we somehow built the joint. So here we have the marginals over Z, and then we have the conditional of x given Z. So the whole thing is a joint model. I don't want to build the joint model. I just want to get one model where I can draw the marginal over x. Okay, so just where I have marginalized out Z. Okay, so this is the joint model is not so favorable for obvious reason, because you have to sample this whole let's say path space of environments, and it's also, if you have multiple cells, then you have to sample a lot. Okay. So how to go about? So I think this is quite obvious for the mathematical audience, but I just thought I make this slide. Is that if I say I want to build a model that behaves as if it would be still embedded into the random environment, it's equivalent to saying that I want to marginalize over the random environment. Okay, and sometimes this is misunderstood by saying I'm averaging the process somehow. So I'm never averaging the process. I'm only averaging the path measure, let's say, or the distribution over the process. And that's what I wanted to say here. So if I want to marginalize out the random environment, then it means that I built the expectation over the random environment of this conditional distribution of x given Z. Okay, so I'm averaging distributions, not trajectories. So it means that every trajectory in the marginalized version is also a plain fine jump process, right? It also means that every process that I can build by drawing conditional, right? If you look on the support of this, for any Z is also possible on the marginal. Okay, so I can draw any of the path in X is covered by the marginal. That's something clear, but I still like that this is Z. Okay. Okay, so then. We have this situation. We have some driving network. In this case, I'm really only considering the one-directional coupling. So, the random environment is somehow acting on the target network, and then one can the general result that we rely on comes from counting process theory from Ott Allen, which says that if you marginalize out this random environment, it turns out that you replace the environment, the link of the environment, by the conditional expectation of the environment given the whole history of your target process. Okay? So it's really only the conditional expectation. This is not the approximation, that's just the result, right? That's how it is. And as you can see, it's still a set sub t, so so the dynamics, the environment can fluctuate. So it means that in general you have to solve some kind of optimal filtering problem. So you are estimating a hidden process set by measuring the observed process X. Okay. Also hidden Markov model type of thing. Just another way of saying stochastic filtering or optimal filtering. And if you think about this, this gives us a way to actually decouple any kind of stochastic network into two parts, where we then say, this part I don't care, I want to marginalize it out. Then I'm replacing any of the inputs from this network to my target network by the conditional expectations of the inputs given the history of the process itself. So I'm not going to talk about the dynamics set in this talk. So this is covered in our Blows computational biology paper if you want to have a look on this. So what I'm considering here is the simpler case if Z is a static but random environment. Okay, so think again of the ribosome example. So let's say that every cell is picking up a random number of ribosomes, and this is somehow assumed to be constant, right? Then if you observe a population of cell, you will see a lot of variability in your protein expression due to the different number of ribosomes, and that's what we want to model. So just an example how this looks like if you do it on pen and paper for simple systems like a Poisson process with a random rate, and let's assume that the random rate is gamma distributed. So then you can actually marginalize this out. You know, at the end, the marginal over x will be a negative binomial with this distribution. But what we are claiming here is that you can replace this, the marginalization, by starting with a marginalized model right away, which is self-exciting. It means that the intensity of the Poisson process is a function of the process itself. Okay. And what do you plug in as a rate? So here I wrote this a bit more complicated just to indicate that this rate, you know, of the Poisson process, that's the random rate. So in the marginalized model, as we said, we replace the real random variable by the conditional expectation of the random variable given the history of the process. So you can see here that I'm just replacing the rate by the conditional expectation. And the conditional expectation of a rate given the history of a Poisson process would be just you count the number of firings of the Poisson and divide it by the time, right? And that's what we do here. So xs is the number of counts of the Poisson process, and I divide it by time. As we are computing a posterior mean here, we also have the heterogeneity given by alpha divided by beta. So if you have no observation given, then the mean, then the rate is given by the mean of the heterogeneity. Okay? And we see that this turns out to be the same distribution, so it works. So in the general case, so as we said, we look on stochastic population models, so for chemical kinetics. So if you have a reaction network where the hazards are given like this, and every of the rate constants, let's say, are assumed to be random, then the hazards in your marginalized network is obtained by just replacing it with the conditional expectation. And the nice thing is that, so this is really an object that is not easy to handle, right? So I can write this down, but by default it's not really useful. But the nice thing is that for some distributions, for some choices of the distributions, you can actually solve this. So in particular, if you again assume the heterogeneity on the rate constant to be a gamma distribution, then you know this is conjugate to the Bass density, and therefore the posterior over so this the conditional distribution of Z given the history is also gamma, and therefore you can plug in the conditional mean being, you know, the first argument of the gamma divided by the second argument. Then you get an explicit form of the hazard for the marginalized network. Okay. Okay. And this is then easy, straightforward to simulate. So you know, you keep on. So Ri is the number of firings of reaction I in the path x, and this is just the integral along the path of my hazard function. Okay. So this becomes just a time-varying propensity, but it's easy to simulate with known algorithm. So we also see that that this system is not Markovian anymore, so it depends on the whole history of the path. But if we now make a state augmentation and say we also keep track of the counts and also keep track of the strange integral, then we make it Markovian, and therefore we can write down a master equation for this as well. So recall that the one way of writing down the chemical master equation is of this form with this step operator. So you know this step operator is shifting everything on the right hand side by minus nu I, and then the new master equation on the augmented state space has here the same jump term, and then in addition it has this transport term here. So it's really type of let's say hybrid master equation as we had before. So we had the discrete jumps, and then we just have a first order derivative. And we have this expression for the marginal hazard for the gamma heterogeneity as we had it before. Okay. Okay, so how can we use this to do some inference, or especially how to dissect this extrinsic and intrinsic variability? And I'm really cutting this short. So we have single cell data where we measure the total fluorescence of the expressed protein. So we build up a hidden Markov model. So the intensity is some kind of a noisy version of the protein that of the protein abundance. And then what we really want to compute is the heterogeneity in this random rates, let's say, or random environment. So if I assume this random rates to be drawn from some parametric distribution that is parameterized by A, what I really care about is estimating A. Okay, so I want to compute the posterior over A given all my data. Okay. So think of A as being the variance and the mean of my rate constants. I want to describe the heterogeneity in my population. Okay. And so you can also map this to a graphical model. So it means that for every cell we have our Markov process down there, and it's controlled by these random rates, which are different from one cell to the other one. And then I'm also spending some rates that are shared across all the cells, because I believe that maybe if it's a basic biomolecular process, then the rate should be a certain number and cannot change from one cell to the other one. Okay. So then we generate this data using some home-cooked microfluidic devices, very similar to what Peter Swain pioneered some years back. So we have these cell traps, and then we are measuring the fluorescent intensity. So the final result is that we can decompose the cell-to-cell variability into these two components, extrinsic and intrinsic, as you can see for a gene expression system in yeast. And if we do then the corresponding dual color experiment, so we insert two copies of the same construct in the gene, and then do the decomposition, computing the two principal components for the dual color experiments, we get a very good agreement to our inference procedure. Okay. Good. So now let's switch gears and look into another system where we go to, let's say, space, and in particular we want to study. We take the system that many people use. It's the MS2 or PP7 system. So what kind of so you insert in your gene of interest, you insert the stem loop sequence, and this to this stem loops, to this nascent mRNA stem loops, code proteins are binding. This could be either MS2 protein or the PP7 protein. If you label this protein with fluorescence, then your mRNA gets decorated while we are transcribing the gene. So while the mRNA is synthesized, the labeled code proteins are binding, and we will see a bright spot in under the microscope if a transcription is going on. Okay. And we make this inducible by using the GEV system, so we can add beta estradiol to the system and then start the expression of this system. So we have the stem loops in front, 14 copies every. So 60 base pairs long, every stem loop, and then we have the normal gene for the guide, the sequence for the guide 10 gene. Okay. So that's our system. So what would you expect? So if you have a transcription event, the polymerase is binding, then it starts to transcribe the stem loop. So you will see an increase in the intensity. Then it transcribes the gene coding sequence, which is not labeled, so we will have a constant part. Here are the stem loops, then the remaining bases are transcribed, and then at one point the nascent mRNA is falling off and it's floating away, and you're losing the signal. Right? How does this look like under the microscope? So I hope one can see something. So here you see the X, Y, and then you see the XZ projection. So we do 9 Z slices, and here you can see the YZ projection, and then you would see some spot appearing at one point, which is a transcription site, a transcription event where you see the lightening up of the single code proteins, and you should see an increase. Okay, and this is just keeping track of the spot in 3D. And clearly, if you have induced the gene, then you have bursts of transcriptions. So new polymerases are jumping on the system. Right. So this is one yeast. Yeah. So we cooked up a tracking algorithm that at the end of the sequence you see it fails, but most of the time it keeps correctly track of the spot. So we corrected for any kind of point spread function and used the information for quantifying the spot intensity and also the position of the spot in 3D. So we measured this with 100 nanometer bits, the point spread function, and also computed this using a theoretical model. And so this we try to correct for this intensity. So then we built a stochastic model for this. So this is really now only the elongation process itself. So so it's more or less a standard TASEP model, so a totally asymmetric exclusion process. So the free RNA polymerase can jump on the gene, and they can only jump first if the first sites are actually free. If there's no other polymerase, then it can move along the gene as long as there is space in front, and then at one point it hits the termination, then it jumps off the gene. Okay, that's the only thing. So that's a discrete. This is a jump process, a continuous time discrete state space. So what is our state space? So we have we start let's say with n polymerases. These can take on either values -1, which means that they are detached, or they take on the different positions along the gene, which for a gene of length L, we have 0 to L -1 such positions. We have n genes, so the state space is just some subset of this Cn. Okay, and then we are looking for the generator of the CTMC that looks like this. So we only have 3 events that we need to care about. So we have the initiation. The initiation is possible if we have one polymerase that is in the state -1, and if the first delta base pairs are free, right? If there is no other polymerase sitting on those things. Then I have elongation moves if I have a polymerase K that is somewhere between 0 and L -2, right? And if in front nothing is blocking the gene, the polymerase. And then at the end of the day, the polymerase can fall off if it reaches position L -1, and it turns back into the state -1 to be a free polymerase. Okay, that's our model. And again, we use a hidden Markov model. So because there is, as you saw, a lot of noise on the data, so you have to build some noise model for it. So what's first our let's say noise-free observation? It's the number of stem loops. So this would be, a stem loop is k bases long. Xi is the position of the ICE polymerase along the gene. So if I divide this, this gives me the number of transcribed stem loops. And if I'm reaching the maximum m, then I cannot go any further. And then the intensity is not increasing anymore because I'm in the gene coding sequence. Okay, so that's and I sum up over all the polymerases. This is giving my total stem loop counts at every time. Okay, and then we can assume a Bosonian observation model. So conditional on my number of fluorophores that I have on my gene, what do I measure in terms of counts? So this could be some scaled version of the total stem loops plus some background current. And this would be then the observation density that I need to carry. And as for discrete time models, or for standard hidden Markov models, the forward-backward recursion is implemented. You can do the same thing for continuous time Markov chains. So what we want to compute is, given those observations, noisy observations at certain time points. So we have, I think, 9 seconds or no 11 seconds sampling rate, these intensity traces, we want to compute back the states, the positions of the individual polymerases. Okay. So this is now a conditional Markov process, or so you can derive a smoothing master equation. So this conditional distribution is also satisfying a master equation. The only difference is that now you have time-varying intensities. Okay, because the process has to be steered to the observation points, and therefore, you have to change the you have some external forcing to drive the Markov process to the observations. And in order to compute this smoothing generator, you have to solve the Chapman-Kolmogorov backward equation with the jump conditions at the observations. So this is just the continuous time version of a standard hidden Markov model, and what it gives you is this conditional distribution. But as you know, we also have unknown rate parameters, so we want to do state estimation together with the rate parameters, and this brings me to the parameter estimation scheme. So what we do is Gibbs sampling, so we can draw conditional on the parameters. We can draw a path that's exactly the posterior path, and then we turn it around and we get we can draw parameters given the path. So we keep on iterating, and we sample from the joint. Okay? And this would be some kind of the traces that we get for the posterior distributions for the number of stem loops on the gene given the observations. The observations are here the crosses, and this is somehow the predictive density. So what's the probability of a new data point given all the other data points? Okay. So how am I with time? So no, I still have 10 minutes. Okay, so I got confused now. Yeah, so the nice thing, again, you can use the Markov property, as I told you before, the conditional distribution of a rate parameter given the whole path for a Markov process is again gamma distributed with only two parameters. The sufficient statistics of the Markov process is just this waiting time and the integral, and therefore you can efficiently draw those parameters. Mm-hmm.
The posterior at the bottom right, they're coming down before the RNA is falling off. Is that the fluorophores coming off, or what?
So here you see a quite potentially complicated composition or sum of multiple polymerases along the gene. So this is perfectly one polymerase being on the gene falling off. This is one, right? But then you see, so in this case we have 24 stem loops. So sorry, that's an old data set, and then so you see one transcription, but then you see another mRNA going on. So therefore you go to double the size, and then you get this strange interleaving. Some is jumping off the gene, and another one is jumping on. Yeah. So the problem with this is that it's very computationally expensive, because to compute this conditional path, this smoothing path, so drawing a path conditioned on the parameters but conditioned on the observations, you always have to solve this backward equation and then compute the smoothing generator for every iteration of this Gibbs sampling. So this is quite expensive, and we thought of something cheaper. And the first approximation that we do is we approximate the elongation process by a deterministic process. So why is this good, or why can we do this? So the standard picture, as you recall, is a linear increase. Then we have a plateau for the gene coding sequence, and one point it falls off, right? So every of the small steps is exponential. So we know that the whole waiting time from here up to here is an Erlang distribution, or from here to here is the Erlang distribution, and then you can see how the variance of the Erlang distribution is scaling with the number of steps. So k is the number of steps, and then you see it falls off with 1 over k. So as you expect, the smaller the steps you have, the tighter the weighting distribution is getting. So here it's already indicated for if you have 500 steps, the deviation to the deterministic path is already very little. We have a gene that is 3,000 base pairs long, so this is quite okay, I would say. But it's still an approximation. Okay. Okay. So what we then do is we have this deterministic model for the elongation. So the randomness is now in the initiation points. So the whole parameter that, if you wish, we have to sample over is the number of initiation points for the n polymerases, and then we have the elongation rate and the termination rate, and we have some parameters for the noise, for the observation density. Yeah.
So you don't think pausing is a big thing?
No, we would love to see pausing or and also stalling, and then this model is not really good anymore. But from the current data situation, I think we are not right at the. We are not yet in a position to really answer the thing whether polymerases are interacting with each other. So I have this exclusion term that they cannot get closer than what is biophysically possible, but this model is not could not account for any deviation from the linear elongation. Okay, so that's our unknown parameters now, and we do for every of the choices of polymerases that we could have on the gene, we do model selection. So we're computing the evidence, the marginal likelihood for it. And in principle, as indicated, it wouldn't be a problem to somehow replace it by the linear elongation by some sample bars, but it's really a complexity issue for now. So what are we using for this? It's so we do MCMC on these parameters, but as you can expect, somehow if you look on the profiles, the initiation points, they are somehow in a multimodal space. You know, if you deviate a bit from the initiation position, there will be very little probability anymore that you will build a good trace. Okay, so therefore we run parallel MCMC and we heat them up, and then we successively cool them down. So something like parallel tempering or annealing of the Markov chains, and then we keep on at every iteration resampling according to the importance weights of the resulting samples. We are resampling the chains. So we would have like 100 MCMC chains in parallel, and then somehow the good chains are surviving as we're cooling down the system. Okay, so it's a bit more in detail. So we have this is the target distribution, and then we have a tempered version. So we take this to some exponent, and therefore it's broader, and therefore it can explore better the parameter space. It's not getting kept caught in some local mode. And then if you keep on cooling down and then doing resampling of the chains, then the importance resampling is just if you cool down the chain from beta to beta plus delta, there's only a very little difference. So this, and if you do this gradually, you can explore the parameter space very well. So the two extreme points are the true posterior. So if you take this to the power of 1, but if you take it to the power of 0, you're just drawing from the prior, right? So the most heated chain would be the prior, or which you assume to be broad and nicely behaved, and then you're cooling down until the posterior. Yeah, that's just one more time the resampling scheme. So you get from every chain you get one sample. Then you see how likely those samples are under the new distribution, which is the cooled distribution. So these get importance weight, and then according to the importance weight, you're resampling the chain. So in this case, the one chain that from theta, which is producing theta 1, if you do the resampling, it will not survive. So you will replicate instead the sample of you replicate the chain 2, right? And then you keep on going with the chain 2. The good thing is that if we have this annealing schedule, we actually have a nice way to compute this model evidence that we need. You record for every of the different choices of the polymerases. We want to compute the model evidence for this. So we want to get the marginal likelihood of the observation given n, the number of polymerases. And so a well-known technique for computing this is thermodynamic integration. So but for this you have to have the expectation of these quantities at different temperatures, and that's exactly what we have for our annealing schedule. So we can take a Monte Carlo estimate of this expectation and get a robust estimate of the model evidence. So how does this look like now for our actual data that we measured in the lab? So this is one exemplary trace. So you see a lot of initiation events here. So this is the reconstructed number of stem loops. Here you see already again very confined posterior probabilities, and I'm also showing you the MAP path, so the maximum a posteriori path in blue. And here I'm showing you the predictive density, which somehow indicates that the samples that we have measured are somehow well explained with our observation model. Okay. And here you can see the posterior distribution over our initiation points, where we say at this point a polymerase is jumping on the gene. Okay. So that's the state estimation, but we do parameter estimation in addition. So what are the two parameters? It's the elongation rate one time and the termination rate. Right? So the elongation rate turns out to be around 30 base pairs per second, and so we're just indicating the gray shaded area to be what people have reported in literature for yeast elongation. So it's in the right ballpark. And we start with a very uninformative prior, so it's a 1 over x prior. So and you see that the posterior is very well converging to the rate. And also for the termination rate, we get reasonable values, also starting with a very flat, in this case exponential prior, which is the, let's say, the prior of choice for a scale parameter where you don't know at which orders of magnitudes it's lying. And so if you compute the total dwell time, you get, like, if you have a 3,000 base pairs of our gene, and we produce around 30 bases a second, then we have around 100 seconds plus a dwell time to do plus a dwell time of 30 minutes, then we get around 30 seconds, then we get about 2 minutes of dwell time of the nascent RNA on the gene, which is again in the right ballpark. So the interesting thing is that everything has done before in terms of the efforts coming from Rob Singer are done on autocorrelation type of analysis. So it's really inspired just by fluorescent correlation spectroscopy. So you keep on collecting a lot of traces at stationarity, computing the autocorrelation, and somehow getting the number of from the decay of the autocorrelation, you can somehow estimate the length or the dwell time of the polymerase of the mRNA on the gene. Right? But here we can use one trace to already get good estimates. So this is ongoing work. So we want to see now, is there heterogeneity from one cell to the other according with respect to the elongation rate and also termination rate or also initiation. Okay, so this brings me already to the end. So I think we need to disentangle the heterogeneity that we see in the data due to effects from the environment and the target network. The marginalization of the process of the Markov process will help for this, I think. And in general, this is a tool to somehow decompose stochastic networks in general. And then I showed you, which is the space part, is that we can do station state inference for single molecule data, which is not restricted to. So sorry, they are missing, especially for non-stationary settings. So which is not restricted to stationarity, which you normally need for any kind of autocorrelation analysis. So thanks a lot.
So to just to understand your last result, so this was just based on the trace you showed, the experimental trace, you just one experimental trace.
Yes, yeah.
That was the, okay.
I was surprised you didn't see any bleaching or include a parameter for Do you have p-loss?
Oh yeah, we have.
Oh, you did?
Sorry.
Sorry, I missed it.
Yeah, yeah. So we have this B, which was a collection of. So we have the bleaching rate, and we have the offset, and then we have, in this case, we have Gaussian. We assume Gaussian observation, so we have the variance of the Gaussian. So that's the 3 observation parameters that we have. Yeah, it's strongly bleaching, unfortunately.
Just to follow up on the one trace. So, in principle, if you have multiple traces, you would want to somehow treat them all together because some of the parameters would be shared between the bleaches, not the on and off places, but things like the bleach rate or the noise. So,
Sure.
Would that significantly help your determination, or it looks like it's pretty clean already?
Yeah, we are also surprised that we can actually on the elongation rate get such a good handle even on a single trace.
I mean, was that trace cherry picked? Like, are some traces a lot worse?
So for some traces, yeah, clear. I think the selection cherry picking was done in terms that, you know, by eye you could see that there is some initiation, some true initiation event going on. So in this sense, we downsampled the number of, but there are many traces that. But this is ongoing work, so we would definitely like to see whether we have heterogeneity. So doing single estimation of the traces and then combining it in this hierarchical model, as I explained at the beginning, that we have maybe one parameter controlling the heterogeneity in the population. So this is not final.
Okay, great. Well, thanks, Heinz.